Hi, good evening, everybody. We're back with our lectures on the general principles of cell communication and molecular biology. Right. Now, the concentration of a molecule can be adjusted quickly only if the lifetime of the molecule is short. It is natural to think of signaling systems in terms of the changes produced when a signal is delivered, but it is just as important to consider what happens when a signal is withdrawn. During development, transient signals often produce lasting effects. They can trigger a change in the cell's development that persists indefinitely through cell memory mechanisms such as those discussed in the former chapters. In most cases in adult tissues, however, the response fades when a signal ceases. The effect is transitory because the signal exerts its effects by altering a set of molecules that are unstable, undergoing continual turnover. Thus, once the signal is shut off, the replacement of the old molecules by new ones wipes out all traces of its action. It follows that the speed with which a cell responds to a signal removal depends on the rate of destruction or turnover of the molecules the signal affects. It is also true, although much less obvious, that this turnover rate also determines the promptness of the response. When a signal is turned on, consider for example, two intracellular signaling molecules X and Y, both of which are normally maintained at a concentration of 1,000 molecules per cell. Molecule Y is synthesized and degraded at a rate of 100 molecules per second. These molecules have an average lifetime of 10 seconds. Molecule X has a turnover rate that is 10 times slower than that of Y. It is both synthesized and degraded at a rate of 10 molecules per second, so that each molecule has an average lifetime in the cell of 100 seconds. If a signal acting on the cell boosts the rates of synthesis of both X and Y tenfold, without any change in the molecular lifetime at the end of 1 second, the concentration of Y will have increased by nearly 900 molecules per cell, 10 into 100 minus 100, while the concentration of X will have increased by only 90 molecules per cell. In fact, after molecule synthesis rate has been either increased or decreased abruptly, the time required for the molecule to shift halfway from its old to its new equilibrium concentration is equal to its normal half-life. That is equal to the time that would be required for its concentration to fall by half of all synthesis if all synthesis were stopped, as shown in figure 1510. The importance of rapid turnover. The graphs show the predicted relative rates of change in the intracellular concentrations of molecules with different turnover times when the synthesis F are either A, decreased, or B, increased suddenly by a factor of 10 in both cases. The concentration of those molecules that are normally being rapidly degraded in the cell, red lines, change quickly, whereas the concentration of those that are normal being slowly degraded, as shown by green lines, change proportionally more slowly. The numbers in blue on the right are half-lives assumed for each of the different molecules. The same principle apply to proteins and small molecules and to molecules in the extracellular space and inside cells. Many intracellular proteins have short half-lives, some surviving for less than 10 minutes. In most cases, these are proteins with key regulatory roles, whose concentrations are rapidly regulated in the cell by changes in their rates of synthesis. Likewise, any covalent modification of proteins that occur as part of a rapid signaling process, most commonly the addition of a phosphate group to an amino acid side chain, must be continuously removed at a rapid rate to make rapid signaling possible. Now, to go ahead, we discuss these molecular events in detail for signaling pathways that operate via cell surface receptors. The principles apply quite generally, as the next example would illustrate. Nitric oxide gas signals by binding directly to the enzyme inside the target cell. The nitric oxide gas signals was just covered. So now I'm going to the next topic, all right? In fact, in the last lecture, the details are there. So just after that would be the continuity now. So we just see the figure of nitric oxide, and then we go on to the next chapter. Sorry, the next topic. The role of nitric oxide in NO in smooth muscle relaxation in a blood vessel, or acetylcholine released by nerve terminals in the blood vessel wall activates NO synthase. And in endothelial cells lining the blood vessel, causing the endothelial cells to produce NO. The NO diffuses out of the endothelial cells and into the underlying smooth muscle cells, where it binds to and activates guanylyl cyclase to produce cyclic GMP. The cyclic GMP triggers a response that causes the smooth muscle cells to relax, enhancing blood flow through the blood vessel. Carbon monoxide, CO, is another gas that is used as an intracellular signal. It can act in the same way as NO by stimulating guanylyl cyclase. These gases are not the only signal molecules that can pass directly across the large target cell plasma membrane. A group of small hydrophobic non-gaseous hormones and local mediators are also enter target cells in this way, but instead of binding to enzymes, they bind to intracellular receptor proteins that directly regulate gene transcription, which is also discussed later. Nuclear receptors are ligand-activated gene regulatory proteins. A number of small hydrophobic signal molecules diffuse directly across the plasma membrane of target cells and bind to intracellular receptor proteins. These signal molecules include steroid hormones, thyroid hormones, retinoids, vitamin D, although they differ greatly from one another in both chemical structure. That's shown in 1512. And function, they all act by a similar mechanism. When these signal molecules bind to the receptor proteins, they activate the receptors which bind to DNA to regulate the transcription of specific genes. The receptors are all structurally related, being part of the nuclear receptor superfamily. This very large superfamily also includes some receptor proteins that are activated by intracellular metabolites rather than by secreted signal molecules. Many family members have been identified by DNA sequencing only, and the ligand is not yet known. These proteins are therefore referred to as orphan nuclear receptors. The importance of such nuclear receptors in some animals is indicated by the fact that 1 to 2% of the genes in the nematode C elegans code for them, although they are fewer than 15 humans. So nuclear receptor superfamily refers to intracellular receptors for hydrophobic signal molecules such as steroids and retinoic acid. The receptor ligand complex acts as a transcription factor in the nucleus. So that is about nuclear receptor superfamily. And going back to the subject matter proper, some signaling molecules that bind to nuclear receptors, note that all of them are small and hydrophobic. The active hydroxylated form of vitamin D3 is shown. Estradiol and testosterone are steroid sex hormones. Steroid hormones, which include cortisol, the steroid sex hormones, vitamin D in vertebrates, and the molting hormone ecdysone in insects, are all made from cholesterol. Cortisol is produced in the cortex of the adrenal glands and influences the metabolism of many types of cells. The steroid sex hormones are made in the testes and ovaries, and they're responsible for the secondary sex characteristics that distinguish males from females. Vitamin D is synthesized in the skin in response to sunlight after it has been converted to its active form in the liver or kidneys. It regulates calcium 2 metabolism, promoting calcium uptake in the gut and reducing its excretion in the kidneys. The thyroid hormones, which are made from the amino acid tyrase, act to increase the metabolic rate in a wide variety of cell types. While the retinoids such as retinoic acid are made from vitamin A and have important roles as local mediators in vertebrate development, although all of these molecules are relatively insoluble in water. They're made soluble for transport in the bloodstream and other extracellular fluids by binding to specific carrier proteins from which they dissociate before entering a target cell. Right? Beside a fundamental difference in the way they signal their target cells, most insoluble signal molecules differ from water-soluble ones in the length of time they persist in the bloodstream or tissue fluids. Most water-soluble hormones are removed and are broken down within minutes of entering the blood. Local mediators and neurotransmitters are removed from the extracellular space even faster, within seconds or milliseconds. Steroid hormones, by contrast, persist in the blood for hours and thyroid hormones for days. Consequently, water-soluble signal molecules usually mediate responses of short duration, whereas water-insoluble ones tend to mediate responses that are longer lasting. The intracellular receptors for the steroid and thyroid hormones, retinoids, and vitamin D all bind to specific DNA sequences adjacent to the genes the ligand regulates. Some receptors, as those for cortisol, are located primarily in the cytosol and enter the nucleus after ligand binding. Others, such as the thyroid and retinoid receptors, are bound to DNA in the nucleus even in the absence of ligand. In either case, the inactive receptors are bound to inhibitory protein complexes, and ligand binding alters the conformation of the receptor protein, causing the inhibitory complex to dissociate. The ligand binding also causes the receptor to bind to coactivated proteins that induce gene transcription. The transcriptional response usually takes place in successive steps. The direct activation of a small number of specific genes occurs within about 30 minutes, and constitutes the primary response. The protein products of these genes in turn activate the other genes to produce a delayed secondary response, and so on. In this way, a simple hormonal trigger can cause a very complex change in the pattern of gene expression. The figure 13, the gene expression is shown here, and figure 15-14 in fact. And 15-13, the nuclear receptor superfamily, all nuclear hormone receptors bind to DNA as either homodimers or heterodimers, but for simplicity we show them as monomers here. The receptors all have a related structure, the short DNA binding domain in each receptor. And the details as shown. The short DNA binding domain in each receptor is shown in green, B, a receptor, and a receptor protein in its active state is bound to inhibitory proteins. The short DNA binding is shown in green, and receptor protein in its active state is bound to inhibitory proteins. Domain swap experiments suggest that many of the ligand binding, transcription, activating, and DNA binding domains in these receptors can function as interchangeable modules. The binding of ligand to receptor causes the ligand binding domain of the receptor to clamp shut around the ligand, the inhibitory proteins to dissociate, and coactivated proteins to bind to the receptor's transcription activating domain, thereby increasing gene transcription. The three-dimensional structure of a ligand binding domain with right and without left ligand bound. Note that the blue alpha helix acts as a lid that snaps shut when the ligand shown in red binds, trapping the ligand in place. So this is how it is. So that's it. And we're on to figure 15-14. Response induced by the activation of a nuclear hormone receptor A, and we see that figures here, the early response and B, delayed response, delayed secondary response. The figure shows the responses to a steroid hormone. But the same principle applies for all ligands that activate this family of receptor proteins. Some of the primary response proteins turn on secondary response genes, whereas others turn off the primary response genes. The actual number of primary and secondary response genes is greater than shown. As expected, drugs that inhibit protein synthesis suppress the transcription of secondary response genes but not primary response genes, allowing these classes of genes transcription responses to be readily distinguished. The responses to steroid and thyroid hormones, vitamin D, and retinoids like responses to extracellular signals in general, and determined as much by the nature of the target as all by the nature of the signaling molecules. Many types of cells have the identical intracellular receptor, but the set of genes that the receptor regulates is different in each cell type. This is because more than one type of gene regulatory protein generally must bind to a eukaryotic gene to activate its transcription. An intracellular receptor can therefore activate a gene only if there is the right combination of other gene regulatory proteins, and many of these are cell type specific. Thus, each of these hormones induces a characteristic set of responses in an animal for two reasons. First, only certain types of cells have receptors for it. Second, each of these cell types contains a different combination of other cell type specific gene regulatory proteins that collaborate with the activated receptor to influence the transcription of specific sets of genes. The molecular details of how nucleotide receptors and other gene regulatory proteins control specific gene transcriptions are coming up soon. So we'll move on to the next topic, that is the three largest classes of cell structure, cell surface receptor proteins are ion, channel-linked, G-protein-linked, and enzyme-linked receptors.